all the NSEJS aspirants. This is your Prasanna Ma'am from NEET World. So yes, in today's session, we are going to deal with two major concepts, one involving the thin layer chromatography and second is about glyoxylate cycle which operates in glyoxysomes. These two concepts and the questions from NSEJS Biology 2022 paper we are going to look into. So without much delay, let's get started with the first question. So here if you see clearly in the question, the adjacent diagram shows chromatographic separation of plant pigments extracted from spinach leaves. The sequence of pigment bands from below to upwards is what? The sequence of pigment bands from below to upwards is what? So here if you see in this chromatographic separation, the name itself is giving what? Chroma means color, separation based on the color of the pigments. Okay, separation based on color of the pigments. But then here pigment bands are being asked and the diagram is given. This diagram depicts thin layer chromatography. Okay, so chromatographic techniques based on the color, it can be paper chromatography or thin layer chromatography. As from the question the reference diagram is given, this reference diagram is what? Thin layer chromatography. So to answer this question, first you have to know what is the principle behind thin layer chromatography, what is the mode of action in separating the pigments using thin layer chromatography. These 2 points should be clear to you better, then only you can answer this question. Look at the theory once. Thin layer chromatography includes an absorbent. Absorbent, it can be made up of the silica or cellulosic layer which is polar. Okay. So first a glass plate can be taken, glass plate or a plastic plate, anything might be. This will be coated with the adsorbent. This adsorbent can be what? Cellulose or silica. Cellulose or silica, what are these beta? Polar molecules, polar in nature, remember that. And then this glass plate or plastic plate which is plated with silica is kept in the oven so that the moisture from this adsorbent will be lost. Okay, it will lose the moisture and the plate will be ready for the thin layer chromatography. Then after taking out from the oven, what happens is we need to mark a point over here and take all the pigments which are given in the question. Okay, all the pigments will be spotted individually on this. All the pigments will be spotted individually on this plate, silica plate, and this plate is kept in a beaker containing mobile solvent. Okay, this plate is kept in a beaker containing a mobile solvent. So this has a mobile solvent. Mobile solvent will be polar in nature. Mobile solvent is non-polar in nature. Mobile solvent is non-polar. Remember that. So when this plate, after spotting out what? After spotting out the pigments, is placed into a mobile phase which has what? Non-polar solvent. When it is placed here, the mobile solvent slowly starts moving all across the adsorbent towards upwards. Okay, so here this is there and at this point we have the spots plotted and the mobile phase will slowly move upwards. Okay, along with the non-polar mobile solvent which is moving all across the silica plate, based on the polarity, the pigments also will be started moving. Those which are more non-polar, along with the mobile solvent, they will move upwards and the band will be appearing over here. Those which are more polar, first only they will stop their movement because they can't move along with the non-polar solvent. Okay, understood? Once again I will brief you the experiment, look into it ma. So first what happens? One glass plate or a plastic plate or any plate is taken. Okay, small glass plate let us assume. Over this glass plate, what is poured? Over the glass plate, the silica is poured, silica or cellulose. Okay, over the glass plate, silica or cellulose is poured out. So, this silica or cellulose is what? Silica or cellulose is polar in nature, polar in nature. So, over this plate, when silica is poured, this is kept into a oven so that the absorbent gets dried. Okay, so this is silica is polar, this is adsorbent. Fine, right ma? So, now this adsorbent after coated with silica, this is placed where? Over this plate, over this plate. This is the first step. First step is clear to you, Nama? Second step is what? Spotting. Spotting of the pigments which are given. What are the pigments that are given in the question? The pigments that are given around chlorophyll A, B, carotenoids and carotenols and carotenoids. These 4 pigments are plotted at one spot. Okay. A margin should be taken at the below and 4 pigments will be spotted over here. Okay. This is the second step. First step is pouring the adsorbent, that is silica, allowing it to dry, keeping in the oven. Second step is plotting, taking a margin and plotting the 4 pigments, spotting the 4 pigments individually. Third step is keeping this into a mobile phase, in a small beaker containing mobile phase. This mobile phase or mobile solvent can be anything like petroleum ether or hexane. Okay, petroleum ether or hexane. So, that mobile phase is poured into a beaker. This is non-polar mobile phase. Fine. This acts as what, Nana? Solvent. Fine, right? So, in this plate, what is placed over here? The silica plate, which is plotted with the pigment spots, is placed over here. So, here we are having the spots. Amongst these pigments, few are polar, few are non-polar. Yes or no? Those which are more non-polar, this mobile phase, as it is solvent, this will move across the polar adsorbent. Polar adsorbent, and this is non-polar solvent. This will be moving across the polar adsorbent. Along with the mobile phase, those pigments which are non-polar will be traversing across the adsorbent to the maximum height. So, those pigments which are non-polar, they will have, the band will appear at greater height. Those pigments which are polar, the band will be appeared in a smaller height, okay, which is nearest to the spotted area. The band will be appeared nearest to the spotted area, those pigments which are polar. Remember that point. This point is clear to you, ma? Here this highest band is appeared, highest band is appeared for non-polar pigments, because they will be traversing along with the mobile phase, non-polar pigments. And this band will be the band which is nearest to the spotted area, this will be polar. Clear, right? So, these 3 points are clear to you, right, ma? Now, after looking into that, now look into theory. Here you should know about one thing, that is retention factor. Based on the value of retention factor only, we will get to know which pigments are polar and which pigments are non-polar, and where we are getting the bands exactly. What is that RF value? RF value is retention factor. Retention factor is equal to distance travelled by solute, distance travelled by solute by distance travelled by solvent. Okay, distance travelled by solute by solvent. For example, the solvent distance is over here and the solute distance is over here. Let us mark it as A and B. Okay. So, here it will be the distance travelled by the solute by solvent. This will give you how much distance is being traversed by the pigment in comparison with the mobile phase. Okay. So, more the RF value means we will get polar band. Less the RF value means less will be the distance between the solvent and the solute molecule travelled. Okay, in that case it will be non-polar. Okay, remember that thing ma, less the RF value it is non-polar, more the RF value the solute is polar. Remember that point. Now you look into theory, look into the theory carefully. More polar pigments are retained more strongly, move least, they will move least and appear at the lower band, and less polar pigments dissolve, less polar pigments will dissolve better in non-polar solvent and move more far, and they will have high or larger RF value. Okay, so relative property of the pigments will be most to least. So here if you see ma, as such when you get the values, the first polar or the lowest band which is occupied is chlorophyll B. Chlorophyll B, when you observe it in the chromatogram, what is the color? It is showing yellow green color. So the first band, that is polar, close to the spotted area, that is chlorophyll B, and what is the color it is showing in the chromatogram? Yellow green color. Most polar due to extra aldehyde group, and it is the lowest band. Second is what? Chlorophyll A. Chlorophyll A is blue green in color. This is slightly less polar when compared to chlorophyll B. So the next band that we are seeing is chlorophyll A after the chlorophyll B. Third pigment, that is carotenols, which is xanthophylls. These are yellow in color. These are oxygenated carotenoids which are less polar, so they travel more height. And lastly it is carotenes. Carotenes are orange in color. Their hydrocarbons are more nonpolar. These hydrocarbons are more nonpolar and they reach top font. Typical RF value of chlorophyll B is 0.25, chlorophyll A is 0.45, and xanthophylls and carotenoids will be more than this, okay, more than 0.5 and more than 0.4. Okay. So here if you see the bands accordingly, accordingly from the question, if you see from 1 to 4. 1 is the lowest value, right? This lowest value will be chlorophyll B, and the next band which is occupied is by chlorophyll A, and then by xanthophylls, and then by carotenoids. So these are the 4 pigments that are being occupied. So option A will be right over here. First lowest band is chlorophyll B, and then it is chlorophyll A, and then it is carotenols, that is xanthophylls, and the highest band which is occupied is by carotene, which is highly nonpolar and it has least polarity. Okay. So here what is the right answer ma? Option 1 will be the right answer. So what are the books you have to refer with respect to the chromatography techniques? Okay, for NSEJS. Best books which are useful and accessible for NSEJS to refer the chromatography techniques includes like NCERT Biology class 11 in chapter photosynthesis during the action spectra and absorption spectra. There the information is given about chromatography, you can go through it. And then this Truman's Elementary Biology is a very standard book, and then MTG, NSEJS, JSO lab experiment section. In this section also past paper solutions, in that also you can look into the chromatography techniques. And HBCSE Junior Science Olympiad experiments notes. Here in this also keenly you can go through the techniques of what? Chromatography. Fine. So this is about the first question ma, where we have clearly analyzed thin layer chromatography. Next question, if you see, this question is on the glyoxysomes organelle. Of course, I am telling you the answer itself, but then you look into the topic. In some plants and fungi, some cell organelles are found which convert the lipids to sugar in the early stages of seed germination. What are these? So, during the early seed germination, fats are converted to sugars by a specialized peroxisome. Those specialized peroxisomes are glyoxysomes, which will activate the cycle called as glyoxylate cycle. Now we are going to look into what is glyoxylate cycle as well. So here if you see ma, what are glyoxysomes? These are the specialized, glyoxysomes are specialized peroxisomes. Glyoxysomes are specialized peroxisomes in which glyoxylate cycle is operated. Glyoxylate cycle is operated, wherein through this glyoxylate cycle what happens? Inside glyoxysomes, fatty acids or lipids, they will get converted to what? Fatty acids or lipids will be converted to sugars. Fine. Let us look into the mechanism in detail, what happens in it actually. Glyoxysomes are a type of specialized peroxisomes. They are present specially in fat storage tissues of germinating oil seeds like what? Castor, sunflower and groundnut, and also in some fungi. Fine. Glyoxysomes are found in the oil seeds and fungi. Okay. So, here the functions, major functions include, they play a key role in seed germination by enabling the conversion of stored fats, that is lipids, into sugars, that is carbohydrates, that can be used for energy and growth up till photosynthesis begins. Till the photosynthesis begins, for the germination process, this glyoxysome organelle will be the chief source in converting the oils or fats into the carbohydrates. Why seeds need them? Okay, because oil seeds will store so much of energy, but that is in the form of fats, not in the form of starch. So, a seed cannot utilize the fatty form, fatty acids form for germination or the metabolic activities of the cell. That is the reason it has to be converted into what? Sugars. Okay. During germination, the young seedlings cannot yet photosynthesize because there is no green leaf in that. It must convert the fats reserves into the sugars, mainly that is sucrose, to support the respiration and biosynthesis. This is exactly what glyoxysomes do. So, here if you see, there are three major steps that are activated in glyoxysomes. Look into the first step. First step is what? Beta oxidation of fatty acids. That is the first step in glyoxysomes. Second step is glyoxylate cycle, which is a modified TCA cycle. Amongst the intermediates of TCA cycle only, there will be little modification utilizing two enzymes like isocitrate enzymes and all, and glyoxylate cycle will be operated with two enzymes utilizing the respiratory intermediates of TCA cycle. In midst you will have two enzymes, isocitrate synthase and malate synthase. These two enzymes will help in conversion of what? Easy conversion of fats into sugars. And third step is transport and operation of what? Gluconeogenesis. Gluconeogenesis is operated at the end. Let us look into one by one. First, beta oxidation of fatty acids. We know fats are converted to what? Fatty acids and glycerol, right ma? So, this is the first conversion of fats, fatty acids and glycerols. Fatty acids will enter the cycle through what? Acetyl-CoA, okay, will enter the cycle through acetyl-CoA in glyoxysomes. This acetyl-CoA, which is the starter of TCA cycle, acetyl-CoA combines along with oxaloacetic acid forming citrate. This is the first step of what cycle ma? Tricarboxylic acid cycle. So, this citrate will be converted to isocitrate by the enzyme citrate synthase. Isocitrate, instead of moving to alpha ketoglutarate, instead of decarboxylating into alpha ketoglutarate, through the enzyme isocitrate lyase, okay, through what enzyme? Isocitrate lyase, it gets converted to, what is the enzyme beta? Isocitrate lyase. Through this enzyme, isocitrate will be converted to glyoxylate, okay, isocitrate lyase. Through this enzyme, isocitrate converts to glyoxylate, and glyoxylate again by the enzyme malate synthase. What is the enzyme? These two are the major enzymes beta, you have to remember for sure. Malate synthase, it gets converted to malate, okay. Now, this malate can operate gluconeogenesis. What is gluconeogenesis? Synthesis of glucose from non-carbohydrate sources is called as gluconeogenesis. Look into the theory once for more idea. So, glyoxylate cycle, this is unique to the plants and this is absent in animals. This uses enzymes like isocitrate lyase and malate synthase. Major functions include this bypasses carbon dioxide releasing steps of TCA cycle and conserves carbon skeleton, converting acetyl CoA into 4 carbon intermediates, that is malate, just by utilizing 2 enzymes and skipping out the other pathways, other steps. Just by utilizing 2 enzymes from isocitrate, which is 6 carbon, by the enzyme isocitrate lyase it gets converted to glyoxylate, glyoxylate converts into 4 carbon malate. Okay? And then here if you see the third step, transport and gluconeogenesis. The intermediates malate and oxaloacetate move to mitochondria and cytosol. Yes, right? Malate and OA will move to what? In mitochondria and cytosol. Through gluconeogenesis, these are converted to what? Sucrose. Through gluconeogenesis, malate and OA will be converted to what? Sucrose. What is gluconeogenesis? Gluco is glucose neogenesis. Neogenesis means synthesis of glucose from non-carbohydrate source. You can take down the definition, beta. Take down the definition. What is gluconeogenesis? Synthesis or production of glucose from non-carbohydrate source. From non-carbohydrate source. Clear, right? Through gluconeogenesis, these are converted to sucrose, which is then transported to growing regions of the seedling. This is the major activity of glyoxysomes that are seen in the seedlings. Okay? So, here if you see the question clearly, here in some plants and fungi, some cell organelles found in which lipids are converted to sugars in the early stages of oil seed germination. What are those? What are those, ma? Those are glyoxysomes. Clear, right? Because inside glyoxysomes, 2 enzymes, that is isocitrate lyase and malate synthase, will convert this OAA first to citrate, citrate to isocitrate, isocitrate from TCA cycle, which has 6 carbon, will be converted to malate by these 2 enzymes, and this malate enters into the mitochondria or cytosol, and this will conduct what? Gluconeogenesis. Malate enters into either mitochondria or cytosol. Mitochondria or cytosol, and it will start what? Gluconeogenesis. Gluconeogenesis. This gluconeogenesis, synthesis of glucose from non-carbohydrate source. Clear, right? Clear with the pathway of glyoxysomes and glyoxylate cycle. Now, next. Okay. And then key features to remember, all the NSEJS recall points. What are the key features over here? We have to remember, beta. Glyoxysomes, peroxisomes. These are peroxisomes in seed. And then they contain 2 enzymes. What are they? Isocitrate lyase and malate synthase, which are the unique enzymes which are seen only in the glyoxysomes, and they convert fats into what? Sugars. Okay, which is most crucial for oil seed germination, and they are present mostly in what seeds? Castor seeds, sunflower and groundnut. These are the main points you have to remember. Okay, the takeaway of this question is to remember these points. So, what will be the best reference books that you can refer with respect to glyoxylate cycle or TCA cycle or with respect to glyoxysomes? These books, okay, NCERT, of course, class 11 biology, where in chapter 14 you will get a clear idea on glycolysis, glyoxylate cycle, gluconeogenesis, okay, and gluconeogenesis. All these cycles you will get a clear idea in respiration and plants chapter, okay. And then this section on fats to carbohydrate conversion in germinating seeds you will get here. and Theis and Ziegler book, which is the standard book for reference of plant physiology. There you can go through the respiration chapter and respiration metabolism and glyoxylate cycle. It will be clear to you. And from Leninger principles of biochemistry also you can get through this glyoxylate cycle pathways. Clear with this right now beta? So, with this today in our NSEJS session we have exercised two concepts, that is thin layer chromatography and glyoxylate cycle in glyoxysomes. In our next class again we will come up with two more concepts, two more PYQs of NEET 2022 and we will be exercising the same. So, that is all for today's session beta. Thank you.